This afternoon, Jesse Cossel, biology major from Marsing, Idaho, and Jennifer Field, biology major from Namp, Idaho, and Brandon Demarco, biology major from Meridian, Idaho. Got that right. I mistakenly said he was from Namp, but he was. You didn't know that. That's crazy. He's from Meridian. But anyways, it's been my pleasure to work with all three of these students over the years. Jesse's been doing some work in Costa Rica ongoing. She got to do probably the more exotic piece of this work, and you'll hear here in a minute where the samples were collected. Jen and Brandon were stuck in the lab, but it was all a good team effort to try to solve this mystery. So they're going to share with us this afternoon the title of their talk, Does Pollution Provide Frogs Protection from the Pathogenic Fungus Trichochytrium denervatus? Guys?
Thank you, Dr. Cossel. We'd like to first start our talk by acknowledging people that helped us with this research. First, the MJ Murdoch Charitable Trust, NNU Science and Math Associates, the Idaho Bureau of Laboratories for their help with our water testing, and then various other individuals for their help with not only the sample collection, but also with the lab methods. So I'm not sure if it's common knowledge to everyone, but frogs are croaking worldwide. 20 years ago, a global amphibian assessment was prompted by the IUCN, and they discovered that 33% of all amphibian species worldwide are in decline or are threatened. And in 2009, the National Geographic posted a post about the different vanishing species, and they posted this picture which depicts some of the biodiversity that has been lost through extinction. And as you can see by this image that's here to depict all the extinctions, not only are there a large number of species that have been lost, but a lot of different diversity among the Endurant family that has been lost. And so these extinctions are happening worldwide and are a serious concern for biologists and people alike across the world. There are a number of reasons for these declines, and here you can see an image provided by the IUCN that depicts some of the factors that include that are influencing these declines. One of the biggest ones here you can see is habitat loss, and some of the other important ones include pollution. And here you can see a bar here, disease. And while disease seems to be a smaller bar, you can see that it is more red than some of the other bars, and that means that more threatened species are being affected by this specific factor. And so disease is a big question in a lot of people's minds about how it's affecting these amphibian declines and what is going on in these influences for amphibians. So one specific fungus that we focused on was the pathogenic fungus Trichochytrium denervatus, also known as BD. And this fungus affects amphibians of all sorts, including frogs that we study. And the first portion of its life cycle is the motile zoospore. So this is a flagellated zoospore that can live in water for 3 to 4 weeks without a host. And after it is developed into a host, it insists in the keratinized structures of its skin, and it goes through a growth and differentiation phase. After this growth and differentiation phase, a discharged papillae forms, and then finally the cap is lost, zoospores are released, and the cycle is perpetuated. Now, it's important to note that an important factor involved in this fungus is the fact that it can live without a host for 3 to 4 weeks in water. This means that it can be dispersed through the ecosystem, not only via host to host, but through the water, where these amphibians live and also reproduce. So because of this factor in its life cycle, it is actually a very dangerous fungus to a lot of Americans because of the fact that it doesn't have to have a host for its entire life cycle. In addition, it does have a very specific temperature. This is 23 degrees Celsius is its critical temperature, or its most abundant temperature in terms of its reproduction and growth. But if the fungus is too cold, it can't grow and differentiate, but if it's too hot, it dies. So the fact that it has a very specific temperature is an important factor in the way that we study it and understand the way it's infecting these amphibians.
So with nearly 2,000 species threatened, we had a lot to choose from, and we decided to focus on a frog that was very charismatic, poorly studied, and also endemic to Costa Rica, which is a place where our lab is very active in research. So we selected Aglathus annie, which is a large highlight frog endemic to the montane ranges of Costa Rica and a small area in Panama. Miss Annie breeds in small bodies, limpid bodies of water such as ponds. And here you can see an image of a male. He's courting, calling at one of our sites. And then this image right here is a breeding pair in amplexus. So Agric Miss Annie has disappeared from most of its former range. This here is a map of Costa Rica, and in yellow you can see where Agric Miss Annie used to be found. And in the center here, this red dot is where it is found today. So these remaining sites are in disturbed areas of metropolitan Costa Rica, so very urbanized areas. And it is thought, scientists think that BD has likely played a role in these declines. Agric Miss Annie used to be so commonly known, its nickname was the coffee leaf frog, and coffee harvesters in Costa Rica used to have to be very careful when harvesting their crop not to kill it because it was found sleeping on the undersides of leaves. And it's also important to note that while Agric Miss Annie is not located in ponds year-round, they spend some of their time in the canopy, and then during breeding season or rainy season, which is our summer season, they travel down and come to the ponds where they're exposed to bodies of water where BD might be found. So with all this in mind, with our disease selected and our study species selected, we developed one major question, which was, does pollution protect frogs from the Trichochytrium dendrobatidis? And we had 3 sub-objectives that we wanted to answer within that question. The first was, does BD occur in Agric Miss Annie populations at disturbed sites? Second was, does study site water inhibit the growth of BD, since we think it might be polluted? And third was just to determine whether or not our site water was in fact polluted. So we had 4 different four different populations in Costa Rica, and this is just a map of Costa Rica. And if we zoom in, you can see, again, I'd like to point out how developed it is. This is all urbanized areas which humans have created for their own use. And our first study site was called Finca Escazú, and this is a gentleman's farm. Actually, it was a man-made pond in which we think he used the pond to mix up his chemicals and herbicides to spray his banana crops with. The water is very inhabitable looking and very hospitable looking. However, we did find what seemed to be a viable population growing there, both calling males and females, as well as successful eggs and developing tadpoles. Our second site was Escazú Fish Pond, and this was actually a series of small ponds located in a gentleman's backyard. And he used these ponds to rear tropical fish for the pet trade industry. And so we can only imagine that since he was located in the central valley of Escazú, that he was exposed to his ponds are exposed to a lot of the pollutants and runoffs. Our third site was Zoo Bolivar, and this is actually our only natural lagoon. All of our other sites were all man-made. So Zoo Bolivar is right in the middle of San Jose, and it's actually an enclosure that the zoo just developed their enclosure around it, and this also houses a site for our Agric Miss Annie, and they seem to be doing pretty well. Lastly, we had Hotel Bougainvillea, and this was 3 ponds located in a hotel's garden, also in a very urbanized area. However, this is, again, a very successful site for our Agric Miss Annie population. So in order to determine whether or not these frogs had BD on them, we had to sample for them, and we had to sample for them during the month of June, the years 2010 to 2013. And we did this by surveying for frogs around ponds and overhanging vegetations at night while they were courting and calling to females. And we swabbed the ventral surfaces of the frogs in order to check for BD using a procedure described by Hyatt et al. In 2007. And so basically we just had a total of 40 swabs that we had to collect with this sterile swab right here. And gloves, different gloves were used for each individual so as not to contaminate each of our samples. And we just swabbed 5 times on each hand, 5 times on the lateral sides, as well as 5 times on the inner thighs, and 5 times on each of their feet. We also wanted to see if there was BD found in the water at each of our sites. So we took samples from 3 different locations of each of our ponds, and we filtered the water through a sterile Stereovex filter. And this was a protocol described by Christian et al. In 2007. And so we drew water up using a 60 milliliter syringe, and then forced it through this sterile Stereovex filter. And this is thought to catch any possible BD zoospores that might be found in the water at each of our sites. So after we were done in the field, we had to bring all of our samples back to the lab. The first step was to extract the DNA from both our filter samples as well as our swab samples. Once our DNA was extracted, we analyzed them using singlet quantitative polymerase chain reaction, or qPCR. And this is a multi-component plot in which you can see the internal positive control in green. And this inflorescence here indicates that our reaction worked, and that if any of our samples contained BD in them, we would be able to detect it. The y-axis is the fluorescence, and the x-axis is the number of cycles it took copying the DNA of BD. So right here you can see it took about 25 cycles to make enough copies of the BD DNA in order to fluoresce. And then in blue, you can see our FAM, or our BD samples. And so the fact that we have fluorescence here after, again, about 25 cycles indicates to us that we did have positive samples in our research. So once we had all the single-kit samples collected, we needed to reanalyze each of them in triplicate qPCR, which we used a known set of standards that we then were able to create this standard curve. And that enabled us to then quantify the number of zoospores found on each positive sample. So we were actually able to know, okay, frog 32 had approximately 60 zoospores on it when we sampled it at that time.
So the third objective was to determine if in fact the site water was polluted. So in 2013, we took a total of 6 liters and collected water at each site. We took 2 1-liter amber glass bottles and collected water for pesticides, and we used water in the amber glass bottles to prevent any pesticide degradation that could be caused by plastic. Second, we took 2 1-liter plastic cubitaners to test for metals, and third, we took 2 1-liter nalgene bottles to do our bioassay trials. And we kept all this water refrigerated at about 2 to 3 degrees Celsius to prevent any degradation of pesticides or metals. After we got the water back, we sent it to the Idaho Bureau of Laboratories, and they ran a series of 3 protocols set by the EPA. There was protocol 200.7, which tests for hardness, 200.8, which tests for metals, and 525.2, which tests for our pesticides. And then to determine if the BD would in fact grow in the site water, we had to grow the BD here in the lab. So we started with JEL 423, which was the culture we chose to use, which is a known virulent culture that does really well in our lab. And we got it from a mycologist who was the one who actually described BD in the early 90s. He was a mycologist from the University of Maine, Dr. Joyce Longcore. After we got the BD here to NU, we cut off 2 pieces of it, put it in a 1% tryptone broth that we put into a 750 ml flask. And about 7 to 10 days of that being stuck in a cooler at 21 degrees Celsius, the phthalate begins to grow. And after that phthalate growth begins, we take it and place it in a 6-well plate with a 1% tryptone agar. We take off half a mil of the phthalate growth in the broth here, put it in on each of the wells in the agar, put it back in the 21 degrees Celsius cooler, and wait about another 6 to 10 days before we're ready to harvest the zoospores. When we're ready to harvest the zoospores, we flood it with broth, take it off the broth, and then put it on a hemocytometer, which is a modified slide with grid squares that is used for counting blood cells. And we use that for, we dye the zoospores and put it on a microscope that's hooked up to the projector, just like you see here, so the three of us can easily quantify it. We use a hemocytometer with the grid squares, so it's quantifiable that way. And then we have a known amount that we're looking for, so we can dilute or we can put more zoospores in it as we need it. And by this time, we're ready to put it in the bioassays on the 96 well plates. And then here I have a video from the Modi cams that we have in our lab here. This here is a phthalate from the part once we're ready to flood the plates and take the zoospores off. These are the zoospores here that are flagellated. This is the infectious stage of the life cycle of the BD that's detrimental to the frogs who are in the water. This is what infects the frogs here. So here we have our plates, and we put sterile water around the perimeter. And the last column here, this is so we can get a zero absorbency reading with the light. In our bioassay trials, then we put a broth in our first column here, so we can get an absorbency through the broth. Next is our positive control, which is broth and live BD, which should grow in the broth just as it did in our, it's the same tryptone broth as we did before. And then our negative control, which is the broth and dead BD, which we hope shouldn't have any growth in it and should be constant. And then our samples, which is the test water and the live BD that should or should not grow. And this is read from day 0 all the way for 8 days, and we read the light absorbency for those days.
All right, so after performing all of our methods and procedures out in the lab and bringing them, out in the field and then bringing them back to the lab, we finally come to our results. And these are the results from our quantitative polymerase chain reaction that Jesse mentioned earlier. And you can see in our first column we have all of the sites listed that we took samples from. And this was addressing, like Jesse mentioned before, this is addressing the question, is BD present in our samples or not? And so here you can see in the second column the number of Agaricus anni samples that we had, and in parentheses next to it, the number of positive samples that we received from each of those groups. And so out of a total of 203 samples over a period of 4 summers collecting, we had 62 positive samples. We were then able to take a mean prevalence for the data that we received, and it came to about 31.5% mean prevalence of BD out of the total number of samples that were taken. Lastly, then, using our triplicate qPCR, which told us the number of zoospores per positive sample that we obtained, we were able to come up with our mean infection intensity, or our average infection intensity. And we came up with a number of about 63.15 zoospores per sample. Now, it's important to note that a clinically infected animal can have a zoospore count of 10,000 or more zoospores. And so while we did have about a third of our samples with positive testing for BD, we only had a level of 63 up, some up to 200 zoospore count, which is actually a very low level of infection for these animals. So that causes us to ask a lot more questions about what this means, why are the BD levels so low, and how are these animals continuing to live with this presence of BD. Second, we come to our preliminary results for our bioassay trials that Brandon went through with you. And like he mentioned, we took absorbance ratings of about 490 nanometers once each day for a period of 8 days. And as the absorbance ratings change in each of these wells, we were able to track the growth of BD by watching the absorbance change. As absorbance goes up, we know that there's more stuff in each of the wells, which is BD. And so you can see here our first column is our positive control, which we used to compare to the samples that we tested. And the next 3 columns are our 3 different sample sites that we use water from. And we chose these 3 sites because they seem to be the most polluted or the most affected water. And so this was the obvious choice to start with our trial. Now remember, we only ran 1 trial this summer, so it was only preliminary. So we weren't able to run statistical analysis on this. But we do have BD growing in the lab, and so for our future work, we hope to continue these trials, run more trials, and be able to determine whether these are statistically relevant results. Next, we got results back from the Idaho Bureau of Laboratories, which did our water testing for us. And remember, we tested for pesticides, hardness, and metal. And out of 102 pesticides that we tested for, all of the analytes were below the protocol detection limits for the company, and in addition, were below, which which are lower than the EPO maximum contaminant levels for standard drinking water. In addition, the 14 different metal analytes that we tested for were also below the EPA maximum contaminant levels for drinking water. So our results back from the lab seem to indicate that our site water didn't seem to be polluted. And so this, again, causes us to ask a lot more questions about our site water and how it's affecting the growth of these animals. So what does all of this mean? Well, first of all, we learned that BD is not prevented from occurring at these sites. We did have positive samples from our frog samples that we took. And so BD is present in each of these sites. In addition, populations of animals were able to persist with these low levels of infection. Remember, clinically infected animals can have zoospore counts of 10,000 or more. And so while there were positive samples, they were very low level infection. In addition, pollution may not be the factor involved with the presence of these populations. Because our site water didn't seem to be overly polluted, it causes us to wonder if different factors may be influencing the persistence of these populations in these urban areas. So for future work, we hope to conduct the additional bioassay trials so that we can run statistical analysis on each of these and understand how the growth of BD is affected by this site water. In addition, we would like to determine whether or not Agolith misani from the Central Valley has developed a resistance or a tolerance to BD. This would involve challenge experiments, bringing frogs into the lab and testing them with BD to understand how they're being affected by the growth of BD or if they are resistant or tolerant to BD. In addition, we would like to explore other factors that could have contributed to the persistence of these populations other than pollution. One of these would include testing the soil around the ponds for pesticides because it is known or it has been questioned whether or not the pesticides could exist in the soil but not necessarily in the water all year round. In addition, we would like to test the pH and the conductivity of the water that we sampled. Another big factor that could be influencing the persistence of these populations is temperature. Like I mentioned at the beginning of the talk, BD is very temperature sensitive. And often urban areas and metropolitan areas have a higher baseline temperature than a forest or a mountain range might have. And so because BD dies at certain high temperatures, we wonder if the growth of BD is inhibited by these high temperatures in the city. Lastly, and of course ultimately, we would love to reintroduce Agolith misani to its former sites and prevent this beautiful frog from going extinct like so many of the other poor souls that have gone before it. So after wrapping up all of this research, some people might be asking themselves, well, why should we care? Who cares if these amphibians are going extinct? Well, actually, there's a lot of big importance to amphibians, both culturally and in terms of ecosystems. A lot of Central and South American countries really revere amphibians, and they often are used as religious symbols in terms of good luck and or fertility. In addition, though, anurians are huge bioindicators. And what do I mean by this? Well, amphibians really affect the ecosystems that they live in. And so, excuse me, when an amphibian species goes extinct, a lot of times their tadpoles were in the ponds containing or keeping the population of algae at a steady rate. But when these amphibians go extinct, a lot of times algae blooms get out of control, which affects the pond life, the fish life, and unfortunately the entire ecosystem in terms of the animals that feed on that fish, and the cycle continues. And so local ecosystems as well as the entire world ecosystem is really affected by the growth and the populations of these amphibians. And additionally, skin peptide research on amphibians has been done recently. For example, one specific skin peptide has been found to be a pain reliever that is as strong as morphine, but is believed to have no addictive qualities. And so different research in terms of the medical field has been done with frogs, and so you can see a lot of different research potential with these beautiful creatures. And so overall, you can see that there's a huge need for understanding more about these creatures, and specifically understanding more about the Trichochytrium dendrobatidis for its effect on these different animals. So we would like to now answer any questions that you have about our research, about this fungus, or about these beautiful frogs.
So, correct me if I'm wrong, you guys said like the optimal temperature for the reproduction is around 23 degrees Celsius.
Yes.
So, one of you mentioned that when you were incubating them, they were at 21 degrees Celsius. Do you think that could have played any important role, things that not optimal?
Probably not. It has a pretty good range of what it can do, and that's not gonna be big enough of a problem. And we were just kind of limited by what we had to store at that temperature. That was as high as it would go.
Yeah, yeah.
And you have to remember too, it's not just sitting in the fridge for the entire time. We have to take it out, test
Right,
it,
right.
and be moving it around. And so having it at 21 degrees Celsius seems to work for you.
Okay.
I can't remember all the places Remar showed that there was BD present, but do we have it in Idaho, and do you have to go to Costa Rica to get it, or is that just, for
No.
kicks?
No, we have it in Idaho, and so the strain that Brandon was talking about, JEL 423, sorry, it's just one of the more virulent. So we determined that in our bioassay trials, we wanted to choose one that wasn't very finicky and could survive in the lab very easily. But yeah, we do have it in Idaho, but I'm not sure whether or not the strain in Costa Rica has been described to be the one that we used in our lab. But that would also be involved in obtaining a culture from Costa Rica and doing all of our trials with the vaguely standing there in Costa Rica. So we don't want to bring it back here, but yes.
So knowing that amphibians populations are going down throughout the world, and especially threatened in tropical ecosystems, how would one select perhaps the species that could be indicators maybe, or those more sensitive or whatever to the fungus?
How can we select them?
Mm-hmm.
Well, we know what not to select. Toads are very resistant towards it. But I would say that a lot of stream-dwelling frogs in Central America have been heavily affected by it, and that's, again, due to the temperature sensitivity that BD has. But it's also more difficult to survey for street frogs because it's just a noisier environment. They're not as big, typically. They're very cryptic. So I think that stream frogs are a really good species to choose.
You mentioned that all the ones that you took had a mean infection, or the amount of zoospores was like 60 on average. Is there like a threshold level that's deemed dangerous? Because it's affecting those frogs, right?
Well, if you look at it definitely does affect the frogs. Now, if you look at the frogs themselves, a lot of the frogs, if you just physically are holding them in your hand, you're looking at them, you might not even know that they're infected. So these frogs with the very low levels of infection aren't showing necessarily large outward signs of being infected. You aren't seeing their posture changing, or you aren't seeing their skin looking deformed. But they still do have BD, and like we mentioned, I mean, it grows in ponds and streams, and obviously that infection could go up at any point, depending on their environment. And so BD is present, obviously, but they aren't looking quite as bad as maybe a clinically infected animal would if you were holding it in your hand. Did that answer your question?
Yeah.
Okay, so Dr. Chen.
Why do you think the Escazú farm had, 50% really high prevalence, but very low count? Is there anything particular? It looked like they had 50% prevalence, but only about 10 per animal. What? I mean, that seems really
You're talking
unusual.
about Escazú Farm?
Yeah.
Well, just one. We only sampled Escazú Farm 2 out of the 4 years of this study, as far as swabbing it. And you can see that we had a pretty low sample size. But Escazú Farm was actually one of our higher elevation sites, so it's possible that that was just a better growing environment for BD, for the frogs there.
But then they don't have very many, so it just seemed like a
so
really,
she's asking about this.
they're really high infection rate, but really low infection intensity. I was just wondering if you had any speculation about why in the world that was, because whatever we do with that water, that seems like, that's great.
Yeah, I'm not sure. And actually, this was the most alarming site, just because the water was literally green and looked very horrible to live in as an amphibian. Yeah, so I'm not sure. I mean,
But it didn't turn out to be more polluted, right?
no, all of them were below the
Below
max
the-
contaminant levels.
Drinking
So-
water.
The world looks different to frogs. And
It does, apparently. I wouldn't swim in it though.
You're no frog.
Actually, maybe
Not
the
yet.
best place to live is at the zoo because you've got low prevalence and intensity, so.
Minus the cane.
And then came in, okay.
Just thinking about the distribution of this particular species and the fact that it's disappeared so much from its range, do you know if there's anybody trying to sort of span out and sample for it in the vicinity to try to find other subpopulations in what hopefully is like a big metapopulation?
Well, there is a small population in the edges of Panama where Costa Rica and Panama connect. But I don't think there's been any other surveys. We have Aglypis annie in our lab and are trying to breed it just to, maintain that that animal success, even if it is in captivity. So I don't think anybody's necessarily surveying for other populations of dogs. But we are active in a location where it used to be found pretty commonly and have never come across it. So,
Do you know
pretty rare.
Sue Bolivar is trying to breed it? No.
These are not, these frogs are just in the lagoon, so they don't even have them on display for people to come look at. They're just in a lagoon that actually houses some turtles, some little turtles,